Okay, I see people logging into the chat room, so we'll wait just 1 or 2 minutes before we get started. Thanks to those who are joining early. See a few more people still logging into the chat room, so we'll just give it about 30 seconds or so more, and then we will go ahead and get started. Here they come. Okay, it looks like it's leveled out a bit, so we'll go ahead and get started. Good morning, good afternoon, good evening, everybody, depending on where you may be logging in from around the world. I would like to officially welcome you all to the next installment in our Lock and Key Immunodetection webinar series. I am Rob Burgess. I head up business development for Sinobiological, and I want to tell you we have an excellent speaker today who is going to talk about biomarker quantitation, multiplexing, and ultrasensitivity. I think you're really going to enjoy this webinar today. We have one housekeeping issue, and that is, first and foremost, I'd like to ask everybody on the call to please just type into the chat box, hello, and tell us where you're from. We're always very interested in where people are logging in from around the world. And then second, if you have questions for the speaker, I would ask you to just simply type those into the chat box as well. And at the conclusion of the webinar, I will verbally run through as many of these questions as I can with our speaker. So please do ask some questions, and I'm happy to get to them at the end of the webinar. And with that, without further ado, it is my pleasure and honor to introduce our speaker today. It is Dr. Scott Crick, who is the Director of Research and Applications at Brightest Bio. Dr. Crick has been at Brightest Bio since its founding in 2018 in this position. There, he has overseen the development of their ultrasensitive multiplexed PFLISA platform, which includes the development of Plasmonic Fluor, immunoassays, hardware, and the corresponding software. Prior to joining Brightest Bio, Dr. Crick spent his career at WashU in St. Louis, where he received both his undergraduate and graduate degrees in biomedical engineering. And this was followed by a postdoc fellowship there in neurology. And then he also spent a few years as a licensing associate in the Office of Technology and Management there at WashU. Dr. Crick's academic research focus was primarily directed to molecular driving forces of protein aggregation and neurodegenerative diseases. The title of Dr. Crick's talk today is PFLISA Ultrasensitive Immunodetection Platform. It is a next-generation high-performance immunoassay platform with incredible sensitivity, multiplexing, and a familiar ELISA-like workflow. Dr. Crick, it is a pleasure and honor to have you today, and I will now turn the chair over to you.
Yeah, thanks a lot, Rob. That's a great introduction, and it's a real honor to be invited to speak today. Thanks to you and the Sino team who've been really good partners with us for a number of years, and really appreciate it. And all the people that tuned in to join and spend an hour with us today, letting me talk about our platform. Thank you so much. So I'll just go ahead and jump right in. Some of you, I think people typically think when they think of ultrasensitivity, they think about detecting things that they haven't been able to detect, things that are too low to measure with the current assays. And that's definitely part of it, and it's an important part because there are a lot of important biomarkers that aren't detectable with standard ELISA or even other high performance immunoassay platforms. However, it also buys you a few other things that I don't think people think about very regularly. And so I just wanted to orient you to those. And then I'll give you some examples later on. So one thing that you can do, aside from detecting the undetectable, is you can serve precious sample. So many times you have samples that are spread across multiple modalities, multiple different assays. Everybody wants a piece, especially for disease samples. You might only have a little bit. So you want to get the most out of that. And what you can do with an ultrasensitive assay, even if you can measure it with ELISA, is you can dilute the sample more. And in our case, you use a lot less volume to begin with, and with multiplexing, you get even more information. So that's an advantage. Additionally, ultrasensitivity translates across the entire range of that curve. Not only can you measure things at low abundance, but that propagates throughout the middle of the curve too. And I'll show you an example of that with air profiles where you actually get more confidence in your result in the middle of the curve. So even if you can measure it with something else, you get a more reliable, robust, and higher confidence in the answer you do get. And so you can resolve concentrations more easily, even in that range where maybe it's not femtogram per mil, maybe it's in the tens of picogram per mil. You can resolve them more regularly. And in many cases in diseases, you might be looking at a twofold difference, and that makes all the difference. And so you want to be able to pinpoint that accurately. And finally, another advantage of an ultrasensitive platform, and in our case, we have a homebrew platform that's super easy to use, is easier assay development. Ultrasensitivity allows you to relax some of the rigor that you might have with selecting antibodies, uh, in allowing you to dilute more, so you might not have as much diluent optimization, so you can get rid of some of those matrix effects. I'm sure many of you have experienced, if you've developed your own assays before, you've tried all of the commercially available antibody pairs, and maybe you still can't detect what you're looking for. You know it should be there, but you just can't get there. So what do you do? Do you go back and start over from the you maybe do custom antibody development? Now you're engineering the protocol again to immunize and screen, or do you just gain 100-fold sensitivity and now you're there? And that's what ultrasensitivity can provide. So I'll walk you through some examples. And again, I'm sure many of you know ultrasensitivity is important. There are a lot of areas where low abundance biomarkers drive clinical insight, and multiplexing is important as well. It's not often the case that a single marker tells you all you need to know about a disease phenotype. In many cases, you might need to measure multiple markers simultaneously. You don't need to measure hundreds all the time, but especially when you're characterizing these diseases, you might need to measure three, four, five, six, and to get a really accurate phenotype. An example in neurology might be for a good example might be measuring neurofilament light and GFAP to see general neurodegenerative processes, and then you can drill down into like phosphorylated tau to differentiate is this frontal temporal dementia versus Alzheimer's disease, and so on and so forth. And so there are a number of examples here where you can see that multiplexing and low abundance biomarkers are definitely important. We've focused our development in inflammation primarily and neurodegeneration, two areas where these two things are critical. So as a quick overview of the agenda today, I'll give you how does this platform work? What is this platform we call PFLISA, which stands for Plasmonic Fluor Linked ImmunoSorbent Assay? What is it? How does it work? What are the parts? You should know all of that by the time I'm done. And then we'll move on to PFLISA FLEX. This is our ultra-sensitive, simple homebrew assay that any of you who are familiar with ELISA will understand immediately. Finally, we have our PFLISA FLEX platform, which are validated multiplex platforms that we sell as a commercial product, and you get the entire kit. And I'll show how those have been used in studying inflammation, comparisons to other platforms, and also some neurology offerings, and again, comparisons to other platforms. And finally, I'll just summarize, and I'd love to hear what comes up in the Q&A. Really looking forward to that. So again, PFLISA, it's powered by Plasmonic Fluor. And I'll talk about what Plasmonic Fluor is in a minute because it's our secret sauce. Some people call PFLISA, when they've seen the data, ELISA on steroids. And that's actually a pretty decent way to think about it. It's got that ELISA familiarity, but it's so much more powerful. It's so much more powerful in terms of sensitivity, in terms of dynamic range, in terms of sample volume, This is a head-to-head comparison of ELISA to PFLISA, same assay conditions, same antibodies, and you can see, it's clear that you get a much more sensitive curve along with an expanded dynamic range. And the sensitivity, the curve scales multiple log orders in both X and Y. And so it's a really sensitive curve. And the general platform capabilities, like I said, not only do you get sensitivity, you get the high dynamic range. So we get femtogram per mil levels of detection for low abundance analytes, high dynamic range, 4 to 5 log orders generally. It allows you to quantify diseased and healthy samples. And I'll show you an example in sepsis patients where that was critically important to have. Multiplexing, the panels, the system's built to offer 2 to 12 plex capability. So really in that sort of sweet spot for your disease phenotype where you really want to characterize, say maybe 4 to 6 analytes really robustly and reliably. Low sample volume. So at the maximum sample volume we'd use is 15 microliters. Of course, if the markers are more abundant, you can dilute that even more. But it's really economical in terms of sample, especially compared to things like ELISA. Highly reproducible. The curve has exceptional CV performance and accuracy across the entire range. We've spent a lot of time making sure that this platform gives you results that you can be confident in. A significant advantage too is a familiar workflow. Anybody who's taken this platform and used it, who's ever run an ELISA, has never even asked us any questions. They get the instructions and they run the assay and they get great results. Not only that, but it fits in with automated platforms, standard plate washers, so it's really easy to adopt. And as I mentioned before, we have a homebrew system that allows flexibility. So in a lot of cases, you want that all of the performance that can be achieved with the standard PFLISA platform, but you want it with your own reagents, your own materials, PK/PD, these sorts of things, and we give you that. And so it's a super easy transition from a working ELISA to what we call PFLISA FLEX. And when we say it's a no compromise platform, you don't have to choose. You don't have to choose ultrasensitivity and limited dynamic range. You don't have to choose quantitation versus multiplexing. You get it all. High dynamic range, ultrasensitivity, multiplexing is very easy to use. And I didn't even mention here the cost. The cost is significantly less than other ultrasensitive platforms, both in terms of capital equipment and consumables. So now I'll talk a little bit about the PFLISA assay architecture, and then it'll allow you to see exactly why this has an ELISA-like workflow and to understand how it works. So as you can see in this illustration, it's a plate-based assay that's a sandwich immunoassay format. And so what we do is we print planar arrays up to 12 plex, and we print every capture antibody spot in triplicate in the bottom of each well of the 96-well plate. And so at the end of the assay, you run it, you add your sample, you wash, you add your detection, you form the sandwich complex, the detection antibodies, and our biotinylated cocktail for multiplexing. And at the end, we add our key, the powerhouse to everything we do, the Plasmonic Fluor. And it's a proprietary reagent that's more than 10,000 times brighter than any existing fluorophore, and it's really the key to our ultrasensitivity. That Plasmonic Fluor, like I said, it's our secret sauce. That's what makes us special. And so it's kind of a complicated structure, but we've been making it reproducibly for years now. We've had three years in continuously where we haven't had a single batch failure. And so we make it at large scale, reliably, and it is amazing. It's an amazing molecule. And so when we use it as a reporter, we get much more sensitive immunoassays. I'll just go through the science briefly. It's a silver-coated gold nanorod plasmonic Fluor. And what that acts as, it acts as an antenna to concentrate incident light that allows us to excite attached fluorophores. Now, these fluorophores, they need to be attached to the particle at a specific distance. So you want them far enough away that they don't experience quenching, which the metals can cause, but you don't want them so far away that they can't realize enhancement due to the surface plasmon resonance. And so there's a critical layer that we call the spacer layer that we coat around this particle. And so these particles are tunable to a wavelength. And when we match the wavelength of the plasmonic fluor to the dye that's conjugated to the spacer layer, and then get that spacing right, we get significant enhancement per particle. And so not only does the fluorescence lifetime of each dye molecule decrease significantly, the quantum efficiency of each molecule also. So not only are we emitting more photons per second, but each excitation also results in photon release as opposed to quenching and other things, which makes these particles extremely stable as well. And finally, we just coat that with a BSA layer that's conjugated to streptavidin through biotin. We can tune the ratio of streptavidin on that surface, and that allows us to have a very low non-specific binding molecule that enhances where we want it to enhance when we want it to enhance. And so again, it's 10,000 times brighter than traditional organic fluors. The array-based multiplexing that I mentioned, we have a piezojet nanoplotter. So this is a non-contact printer. We print everything in triplicate as shown in this microarray design for our 12-plex inflammatory panel. Every single spot that we print is imaged. We image every well. There will not be a plate sent out that we don't know exactly where every single spot on that plate has been printed and QC'd, which is incredibly important for our robustness. And finally, we actually use that QC image in the post-assay analysis so that we can identify every spot precisely when we extract the fluorescence intensity. And so the way this works is post-assay, we get fluorescence intensity from an individual location. That corresponds to the concentration. We have every plate has a calibration curve. Every unknown we extract the intensity, put it into the calibration curve, and get the resultant concentration. We do have to have a high resolution image of every well in that plate. And so we created a very inexpensive, very compact reader that's a single button. It's shown here. It can easily fit on any bench under the first shelf. We don't have to have anybody set it up. We ship it in a crate. We've never had any issues. It's plug and play. You take it out of the box, you plug it in, you run the QC plate that we provide, and then you're ready to go up and running within 30 minutes. The software lives entirely in the cloud. You can plan your experiment, build your plate map, monitor the status of the readers, all from your phone, from your desk, no problem. It's designed to support 21 CFR compliance for those clinical tests with absolute traceability and immutability of the data. It's super easy. We spend a lot of time, and we ourselves have used a lot of different platforms, and software can be a real pain. And so we've developed it all in-house. We have our software team on site. The analysts and experimentalists have talked to them continuously throughout the years to modify this software to be really easy to use, really intuitive, and really robust. And so immediately, it's easy to create your plate map within seconds, actually. We've got a video on our website, brightestbio.com. You're more than welcome to go check it out. You can get immediately from the analysis heat maps for every analyte so that you have a sense of, did this experiment actually work? Do I see differences in my cohorts just at a very gross level? You get the standard curves, the fits, the back calculated concentrations automatically. And the resultant data output files are easily importable with the raw data and the back calculated information into many different LIMS systems. Now I'll move on to discuss this homebrew assay that I've discussed, the platform itself and how that all works. And so what we call this assay is PFLISA FLEX. And the reason we call it that is because it's super flexible. You can use your own reagents to make an ultrasensitive high performance assay. So it's simple. And as I'll show in the next slide, the assay development, the assay process, it's exactly like ELISA, except instead of adding streptavidin HRP, you add streptavidin Plasmonic Fluor and read on our reader. And so if you've ever run ELISA or developed an assay in ELISA, no sweat. The advantage is, of course, you get the PFLISA performance. And this is real data, buying an off-the-shelf antibody pair, running two assays side by side, first time, coding plates side by side, running the assays side by side in the PFLISA FLEX format and the ELISA Live format for something as hard to measure as human IL-1 beta. And you can see you get significant enhancements in the performance. And so this is non-optimized. This was using the vendor recommended ELISA protocol and reagents, all apples to apples comparison here. Like I said before, PFLISA FLEX, it's super simple. It's one swap in that reporter molecule step compared to standard ELISA. Same workflow. You coat the plate, you block the plate, you add your sample, you add your biotinylated detection antibody, as long as whatever your biotinylated detection molecule is, as long as your detection molecule has a biotin on it, we can read it. So the advantage, of course, is it uses less sample, higher sensitivity, higher dynamic range, but no difference in workflow. So it's that ELISA simplicity with that PFLISA performance. And I should mention too, it's flexible in the way that you use it. It's versatile. So it need not be a sandwich immunoassay necessarily with an antibody coated on the plate. It could be perhaps your antigens coding. It could be a competitive ELISA format. As long as if you can do it in ELISA, you can do it in PFLISA FLEX. Again, with the additional performance benefits. And so I didn't just pick human IL-1 beta. We've done this now for a lot of different antibody pairs. We've done it for some PK/PD work. And again, you see enhanced performance without much complexity involved. And so all of these were, just like I mentioned for IL-1 beta, off-the-shelf reagents, run side by side, same conditions, same antibody pairs. And the performance, sort of speaks for itself. And so again, if you need to quantify something, and say IL-6 is a perfect example. A lot of people are familiar with IL-6. Maybe in healthy samples, you're hovering right between that 1 to 10 picogram per mil range. You're trying to distinguish things. ELISA is not going to get you there. But PFLISA FLEX, super sensitive throughout that range, and you can dilute your sample more. So I mentioned this when I said the dilution, you can dilute it more. You get into that sweet spot range for the PFLISA FLEX assay. This is a highly reproducible assay. A lot of these samples we spike into a range that's measurable in both ELISA and PFLISA FLEX. And you can see again and again that you have highly reproducible data. And that's not, I mentioned before that this sensitivity propagates throughout that curve. There's a really wonderful paper from a group at Stanford talking about comparing assays and a metric for doing so. It's called the resolution of molecular concentration. And I created it here for the PFLISA FLEX for neurofilament light, a marker involved in neurodegeneration. And you can see, this metric provides you a way to assess how confident you are in your ability to distinguish differences in concentration between two samples. And so what you can see on the y-axis is the smallest resolvable fold change that you can measure between two samples using one assay versus the other. So in this case, I plotted it for the conventional ELISA assay versus the PFLISA FLEX. Same antibodies, same everything. And what you can see is that really where a lot of those samples lie for normal humans, for example, that aren't involved in neurodegeneration and have an average age, the sample might be around 8 to 10 picograms per mil, let's say. And so you can see that's exactly where the NFL curve is the most sensitive. In fact, you can resolve changes of only 25% between samples over a range that's more than 2 orders of magnitude, almost 3 for the NFL assay. Whereas for conventional ELISA, that range is much narrower. In fact, it can't resolve that difference between two samples over any of its range. And so that's just an example, too, of what ultrasensitivity buys you. It buys you confidence in your result, even if you don't need to measure it in the femtogram per mil level. And so again, here's another way to look at the data you might obtain from your assay. And so this is an error profile measurement. I've done it in multiple observations, Monte Carlo simulation, back calculated error. And so that window is a 95% confidence interval with a plus minus 20% reference in the dotted lines. And you can see, again, especially in the range where samples actually live, so that 1 to 100 picograms per mil in blood, you have incredible confidence. And it starts to deteriorate, obviously, as you approach the lower limit of quantification. But this is an example where you don't need to measure things at femtogram per mil. You need to measure them really reliably at 1 to 10 picograms per mil, 10 to 100 picograms per mil. And that's another thing that ultrasensitivity gives you. Again, it gives you that confidence in those results. So I want to just change the thinking a little bit, that ultrasensitivity doesn't just mean you're only using it when you need to detect something that you can't detect with ELISA. There are other advantages too, and one of them is more confidence in your result. We have two platforms. It's the same architecture, essentially. The Flex, we give you a plate that you coat yourself. It's got a proprietary coating that allows you to bind whatever reagent you need, and it binds it in spots that are printed on the plate, and the rest of the plate's blocked. In the PFLISA FLEX system, we actually give you a kit. And so we print the antibodies in the array, and we provide you all of the reagents that you need. And so you can see Flex versus Plex, you get very similar results across the board. And we've measured concentrations of samples in one versus the other, and they're identical. So move on to talk about that Plex platform here. Like I said, it's a multiplexed immunoassay platform. It's ultra sensitive, and we spend a ton of time rigorously testing and validating these assays. And so we design them in plexes from 2 to 12, depending on the biological relevance of why you might want to measure those molecules together. I should point out that we can actually do more than 12, and we don't. The reason we don't is that it's very hard to make a really reliable, robust assay with single-digit CVs, highly accurate across more than, say, 12 markers. It's very difficult to make it with 12, and it took us a long time to do it. But I would want to say that we focused primarily in that 2 to 12 sweet spot. And one of the reasons is that we demand highly accurate, highly reproducible assays. And so it becomes more and more challenging to get that when you increase the plex. Again, you get all the plexing, you get the validation, but you still, again, have the performance. And so these are standard curves taken off our software directly. This was an assay that wasn't even run by us. It was run at UC Irvine by one of our early adopters, Dr. Abraham Kavi, who is the Director of Innovative Laboratory Diagnostics. And he's had the system for a couple of years now. And he applied it to sepsis, and he was gracious enough to share this data and some sepsis data with us, and allow me to present it today. And so again, these aren't assays that we're running in-house under ideal conditions. This was run by someone else in their lab. And again, you can see these curves are ultra sensitive across all 12 markers, wide dynamic range, high performance. And they needed that performance because they were measuring sepsis, septic patient samples. So they had 100 septic patient samples, and they measured 97 healthy controls. And across all 12 markers, these are the results. And so the idea is, what's upregulated, what's expressed, what do you see in the blood in healthy versus septic patients, and can you use that to discriminate things? There's a bunch of data that they and insights they made with this panel with regards to gram negative, gram positive, mortality prediction. I'm not going to present all of that. I just want to show you an example across all 12 markers, how they were able to use this platform to distinguish, differences between these. And what they found was actual significant differences in a number of biomarkers of relevance in this case. Sepsis is a pretty extreme condition where the values can change by orders of magnitude, and you see that for things like IL-6, IL-8, IL-10, where that wide dynamic range really helps. Let me focus, though, on one molecule in particular. This molecule, IL-1 beta, has been classically very difficult to measure in a number of different assays, and I'll show some more information about that in a second. But this is a perfect example of how powerful PFLISA can be. And so this molecule in particular is it requires the wide dynamic range. You can see the results. They vary by orders of magnitude. In fact, nearly 5 logs of magnitude that these samples varied in concentration. But if you look at the mean concentration, almost all of the samples are actually below 1 picogram per mil. In the healthy, they're below 100 femtogram per mil. And so you really have to have that ultrasensitivity. Not only that, to be able to distinguish these samples at that level, where you're talking about means that maybe go from 700 femtograms per mil to about 75 femtograms per mil, you really need that accuracy and that precision, because without it, you wouldn't be able to make these distinctions. And so with the traditional assay, you might be able to measure some of these samples for IL-1 beta, but it's only going to be diseased. And so we actually did a comparison to another high sensitivity platform, MSD, and this is their VPlex system. And they have a 9-plex pro-inflammatory panel that we compared against. And what you can see is for the markers, and I don't need to show all 9 that were the same between the panels. I'm just showing a couple examples here. For the markers that were the same and quantifiable, we did get pretty good correlation. So there, if you've got an MSD system and you've characterized your samples on that platform, you can be pretty confident that they're going to translate to ours. I should point out that there is one exception, and that exception is interferon gamma. And what happened on the MSD platform is that it measured 10 times higher than ours. And when we went back and looked at the validation data, we realized that there was a difference in the comparison to the WHO standard. So our 12-plex assay, every single marker is calibrated against the WHO standard with no correction factor. So if you measure the standard on our assay, you'll measure exactly what the WHO reference standard provides. But on the MSD VFlex assay, for interferon gamma in particular, they measure the WHO standard at a factor of 10 higher than us. And so that actually gives you, it doesn't mean they're wrong. They detected a lot. I mean, it's just a way to compare platforms, because as many of you may know who've worked in this world, you might get very different answers among different platforms. And it is nice to have a reference standard. I wish all assays had reference standards. And so I would say that our assay, we worked and made sure that it's exactly measuring accurately the reference standard. But I should say then the other part here is MSD VPlex couldn't detect everything. And so the markers that it couldn't detect, IL-1 beta is a good example of that. Like I mentioned, it is very difficult to measure on a number of other platforms. And this is an example of that. So they couldn't detect more than 11 samples that were measured on both platforms. And, that's an example of, in the septic patient case, you can see in the MSD example, their LLOQ isn't high enough. It's 1.292 picograms per mil. And so in that case, they wouldn't have been able to measure the majority of the samples in that assay, even in that experiment, even with the septic patients. And it's not just the VPlex. They also have an ultrasensitive system that they can call the S-plex. And we did a head-to-head comparison with that system, 40 healthy donor samples. And again, we see that with the PFLISA system, we have much better quantifiability of the samples over the range, whereas SPLEX had significant difficulty quantifying those samples. And so between work we've done and other early adopters of our platform, this shows analyte detectability in PFLISA versus other platforms across multiple different studies with early adopters. And I can't share all of that data, nor do I have time to, but this is basically what we've seen. As you saw in the septic case, we have excellent quantifiability and detectability across all markers in multiple samples. Olink for the comparison in their target 96 panel did not detect quite a few markers that we could. And some of them, like IL-1 beta and IL-4, that are particularly challenging and in low abundance, they don't even have. MSD-Vplex is not quite as sensitive as the S-Plex, as you can see, significant problems with detectability and the number of biomarkers. S-Plex is better. They don't have all of the things we have. But again, IL-1 beta is a significant problem. So the panels we have, so we're a younger company and we're working diligently to expand our offerings. Panels we have now and that we're working on are shown in this slide. So a number of human inflammatory markers. These are all human. We'll work on developing mouse markers. Hopefully later this year, we'll start to develop those. We focus primarily in human markers. So you see it covers a fairly wide swath of especially low abundance inflammatory markers, but also some higher abundance chemokines and cytokines. We've developed these in a way to to make sense. Angiopoietin 1 and 2 ratios matter. For example, PD-1, PD-L1, and oncology is important. And you can see we've got other panels in development there. I'll move on to, I mentioned we focus primarily on immunology and also neurology. This is an example of two molecules, neurofilament light and GFAP, that are significantly, like they're really sensitive biomarkers of neurodegeneration. However, they're not terribly specific, but NFL and GFAP, if they're high, you likely have a neurodegenerative event going on. We have incredibly sensitive curves for both of them, super wide dynamic range. The leader in this field with their ultrasensitive platform for a number of years has been Quanterix. And so we actually performed a head-to-head comparison for NFL and GFAP on 158 samples that had already been measured on the Quanterix platform at Quanterix. And the samples were shipped to us, and we measured them head-to-head. And what you can see is that we have excellent agreement with the Quanterix platform. So not only is it well correlated, but that dashed line is one-to-one agreement. So if you measured a sample on the Quanterix platform, you'd likely get within the same answer on our platform. There's a slight bias for GFAP, but again, excellent correlation between those. And I should mention it's at a significantly reduced cost in terms of capital and consumables. So same answer, high performance platform. We've also moved into, with the AD specific panel, brain-derived tau and p-taus help differentiate from AD from other neurodegenerative diseases. If you're in the field, many of you are aware of brain-derived tau and especially p-tau 217. We've got a triplex assay that measures all three. And in testing on normal human samples, we have 100% endogenous quantitation across all three markers. There's some more work going on from early adopters with very compelling data, but I don't have the ability to share that information. But this is a really exciting panel that we're happy to offer, and I think can be powerful particularly in Alzheimer's disease research, but also neurodegeneration. The product menu for PFLISA FLEX, as mentioned, so this isn't just neuro now. There's other panels, of course, that are there that you saw. We've got it in several different flavors. So one, you see the neurodegenerative markers, NfL-GFAP, the threeplex I just showed for Alzheimer's disease, but also developing TDP-43 and phospho-TDP-43, which is involved in frontotemporal dementia. And there are a number of other panels that involve other low abundance biomarkers like IL-33, IL-17A, IL-22. And one of the things we're working on developing as an entirely configurable system, especially for inflammatory markers, to allow you to choose which analytes you want in your assay. And so we'd like to create a menu of maybe 15 analytes, and it's just pick your plex basically and pick the analytes you want. And this is in a validated format. So you can be guaranteed to get the same performance you'd get in any plex across that panel. And I think that's something that's unique and that's desirable for a number of researchers. So again, I wanted to save plenty of time for questions because that's what I get the most out of this. And I really want to hear from the audience. But again, the summary is we've got an ultra-sensitive multiplexed platform that can help researchers measure what they can't measure currently. And so it's, like I said, it's ultra-sensitive, high dynamic range, reasonable plexing, that's really in that sweet spot 2 to 12, low sample volume, so conserve your precious samples, highly reproducible, the familiar workflow. So if you can run ELISA, you can run this no problem. And then that flexibility of PFLISA FLEX. So whether you're developing your own antibodies in-house, you're working on developing a new assay that you can't find commercially, and you're sourcing antibodies from vendors like Zyno, and you want to build this specifically in-house, and you need that performance that PFLISA provides. The PFLISA FLEX platform is something that I think can help a lot of folks. And again, you get all of this without compromise. You don't have to choose sensitivity, dynamic range, plexing. You don't have to trade off one for the other. You don't have to trade off data depth, and you don't have to spend an arm and a leg to get this platform in your lab and start making measurements today. And so with that, I'll wrap up, I think, and love to hear what the audience has to say and see what if I can answer anything and see how it resonated with folks. Thank you.
Great.
Thanks, Sino. Thanks, audience.
Thank you, Scott. That was a fantastic webinar. We appreciate it. Very cutting edge technology, intellectual property there, ultra sensitive biomarker detection and multiplexing. We just love it. I would remind the audience, please do ask some great questions in the chat box, and I'm happy to get to them. We do have our first question today, and it's from Xiaopin Zhang. So Xiaopin is asking, do you have special amino acid or tag sequences for the label in the assay? Can you elaborate, Scott?
Yeah, no, there's no specific tags. So for this, for the multiplexing, we print everything. And so the capture antibodies are localized to the surface via non-specific adsorption, if I'm understanding the question correctly. So it doesn't use any sort of tagging to localize the antibodies to the surface. For the PFLISA FLEX, it also uses, it's a single plex assay. And so you coat your wells the same way. You don't have to modify your capture antibodies. You coat the plate as you would a standard ELISA. So there's no tagging required to localize the antibodies to the surface. I hope that answers the question.
Yeah, and per the PFLISA FLEX, if I pronounce that correctly,
Yeah,
since you brought it up, I'm sure you mentioned this, but can a user use a standard reader for that, or do they still need to buy the Brightest Bio reader?
Yeah, we get that a lot. No, you still need to buy the Brightest Bio reader, and the reason is multifold. One is that you actually do get, as I mentioned in the presentation, we actually print high binding elements. I can't go into that proprietary information right now. I'm happy to do it once all the IP is secured. But we actually print high binding molecules on a blocked surface. And so the capture antibodies bind to 3 specific locations in the well, and they're localized there into a spot that's fairly small. And that also helps increase the sensitivity of the assay. And so when you coat the plate in ELISA, and then you're reading bulk absorption, when you localize all of the signal to these spots, everything's concentrated there. And so you get a significant enhancement in sensitivity just from that step as well. So the Plasmonic Fluor does most of the work, but you get significant sensitivity improvements from that localization too. And standard readers won't allow you to resolve those spots accurately, especially if they're just measuring bulk fluorescence. So you really do need our reader for that.
Yeah, razor blade business model. Nothing wrong
That's
with
correct.
that.
Yeah.
Next question is from Brian. I'm sorry, Brian, I can't see your last name because it's just too bright in this hotel room here. It says, I may have missed for this system, what machine do you use to measure the plasmonic fluor signal? So I'm assuming that's your reader, right, Scott?
That's correct. It's our Discovery reader. And so it's a one button use reader. You press the button, you eject the plate loader, you load the plate, and you press the button, and you go walk away and have a cup of coffee and see the results at your desk. And so it takes high resolution images of every single well. And it's a very inexpensive system compared to other ultrasensitive assays.
Sounds good. And Brian had a follow-up question to that.
Sure.
He says, plasmonic fluor is so bright, could you also use it for flow cytometry?
Yeah, we get that question too. Yeah, there are lots of applications for the Plasmonic Fluor. The problem for us being a small company is we had to choose one. And it's difficult to support all of these different applications. And so we've chosen to create this ultra-sensitive, highly multiplex platform where we can control everything. We don't necessarily have control over everything in flow cytometry and supporting development and stuff. And so, yes, it could be beneficial there. It could also be beneficial in immunohistochemistry applications, but it's not something we support, and we don't. Right now, as we got to start somewhere, right? And so, as we grow, that's something that we can potentially endeavor upon. But right now, we don't support that.
A lot of other potential applications there.
Absolutely, yeah.
You had mentioned immunohistochemistry, which I think is a good follow-up to a John Andrews question. And John is asking, have you tried PFLISA for measuring antibody binding to cell surface receptors?
Yeah, that's exactly the follow-up question. And the answer is no, not us. So the technology was invented at Washington University, and we have an exclusive license to it. Some of our chief scientific officer and inventor of the technology has done a lot more of these exploratory types of work, having invented the technology in his lab and explored that. It's not something that we've pursued because, again, just the breadth of that space and like the receptors and tissue types and how you deal with the sample is not something that we've really explored, and it's a rabbit hole to go down. But technically, it should work pretty well, I would think.
Yeah, sounds good. And I think I think maybe you mentioned this, but I just wanted to drill down on it regarding the price points of both the consumable kits, I suppose, and then the readers. Can you compare that to what else occurs, like the more standard systems in the industry?
Yeah, so it's going to be a little more expensive than a standard ELISA because of what it does. But we've really tried to price it similar to, a multimodal plate reader, a simpler one, and not, not going to give the price away. We'd like to talk to folks that were still early, but I can say compared to a couple of the other platforms, it's there are multiples higher, many multiples in some cases. So we've priced it to be like you said, this razor blade model, we really want to drive consumables and volume and democratize the measurement of a lot of these molecules because we've talked to a lot of folks that frankly just can't afford to measure them. And that's
Yeah,
unfortunate, really. They can't afford the capital equipment and academic labs especially, but even, CROs and pharma, they care. They care about that. They care about the space it takes up. And so it's important for us to be able to meet those needs and allow people to make those measurements that are really important and can help us all. So
right,
that's my
right. Sounds
answer to
great.
that. Yeah.
Audience, we have a few minutes left. If anybody else has any questions, I guess related to that, and maybe you can't share this, but can you tell us like how many instruments you have out in the field worldwide right now, roughly?
Um, uh, yeah, it's on the order of tens. And
Mhm.
so, we've to be honest, Rob, and I'm sure you appreciate this, as a smaller entity, don't want to spread ourselves too thin. So we want to understand. So we've had a few early adopters. We've got folks in major pharma. We've got some CROs. We've got academics, and so it's spread across, those different domains, and they all have different sensitivities and different requirements, and the software lives in the cloud, and they all have different, issues there. And so uh, we want to provide a great experience for everyone. And so we wanted to understand that um, uh, all of the nuances there before really
Right.
launching fully. And so we've not really marketed the platform yet. This is kind of our first public facing presentation and webinar, in fact.
All right.
Uh, and so, we wanted to make sure all the kinks were worked out and that we have the infrastructure in place to support the demand that I hope is there. And so, we have enough instruments in the world that I think we have a good understanding of what the demands are and that we can serve these different domains because, as I'm sure Sino knows, supplying to all of them as well and supporting them, they are different. CROs versus academics versus major pharma, and so we're confident we can do that now. And so now
Right.
we're ready to open it up, I would say.
Well, that's great. Well,
Yeah.
we're very honored to be part of your initial launch and
Yeah.
announcement, I guess, if you will, of your technology and your
Yeah. Thank you.
offering.
Yeah.
We feel very privileged to do that. And then related to that, it's weird that these questions keep following on very appropriately. Brian's son, I finally could see Brian's last name here, is asking, what is your relationship with Sino? I think you know that a little better than I do. So you want to elaborate on how our companies interact?
Yeah. So, to be perfectly honest, we have a number of suppliers. So Sino's not our, uh, only supplier. They are a trusted partner for some antibodies we use in our assays. We've also engaged with them to develop custom antibodies when we couldn't find some for some markers that we think are important, and those assays are in development now. So one of our trusted vendors, and we value their antibody quality, but again, they're not our sole supplier because it's hard to find antibodies sometimes that it's a lot to ask for one vendor to have
Right.
all the antibodies, but they're one of several of our very trusted suppliers that we have. Yeah.
Appreciate that. We're not your sole exclusive supplier yet. Yet is the key
Well,
term.
yeah, well, let's see. There are a lot of markers in the world to measure.
Yeah, well,
Yeah.
if we don't have it, let us know and we'll make it for you. How's that?
Yeah, some of the acquisitions that Sano's made in the past, so like the phosphorylated antibodies with the Signal Chem acquisition has been important for us, and so getting
Great.
highly reproducible phosphorylated standards where these molecules are very hard to measure, that's a very important thing for us, and we've been really pleased with that, and so. Yeah, it's interesting. Small world, because we
Yeah.
used the Signal Chem before the acquisition, and
Mhm.
and I think Sano cares about quality and the same stuff we care about, reproducibility, robustness, and so they're a good vendor for us.
Great. Thank you. Yeah, it was a natural acquisition target for Sino. It's been very successful for us. really strengthened our portfolio. So we appreciate the kind words about Signal Chem. We'll finish up there. One more question. This is perhaps the most important question of the whole morning, and that is do you have any relation to Dr. Francis Crick?
I wish. I've searched ancestry.com and I can't say that I do, unfortunately. Other
You
than
knew I was going to ask that.
yeah, my family does trace back to Britain, but that's where it stops. So
Yeah,
unfortunately,
no
maybe a
worries.
great relative, but other than that,
No worries. You're still a brilliant scientist, and we sure
thank you.
appreciate your time today. So
Yeah,
thank you with that. And we'll go ahead and wrap things up. I just want to thank all the attendees. We had a great turnout today, folks from really all over the world, India, Nigeria, Italy, Uganda, and certainly all across the United States, including
wow,
Houston, and of course, St. Louis, Irvine, and Boston, and a few other places.
wow.
Scott, I really want to thank you as well for agreeing to give this webinar and executing it so well and telling us about Brightest Bio and your technology. It's just fantastic stuff y'all have going on.
Yeah, thank you. And if anyone on the call, feel free to reach out to me on LinkedIn and connect and reach out to me directly via my email. It's scrick@brightestbio.com. I'd
Mhm.
love to talk to folks and see what they're working on. It's exciting stuff. People are doing such amazing work, and I'm so happy to hear that I could maybe support it. Like when someone gets data on our platform, I get giddy. It's why I do what I do. It's so much fun and so rewarding. Thank you.
Yeah. So yeah, audience members, please reach out to Scott directly at his email if you have any questions or want to collaborate or purchase a system. And then finally, I just want to thank my colleagues, Ivana Ramirez and Natalie Chow, for once again flawlessly executing a lock and key webinar. We appreciate your efforts. They are much appreciated. And then with that, I will say good evening and good night to everybody, and thank you once again.
Thanks a lot. Take care.
Bye-bye.